Thank you to Sheba and PJ for organizing this wonderful symposium and inviting me here to speak. And for all of you for showing up, I love to, okay, actually, let me just make sure I know how to go forward. We're good, okay. I love to talk about the brainstem because I don't really need to introduce it or say anything. Everybody knows about the brainstem. Everybody knows that it's important. We just have this intuition about, we need it to stay alive. There's this really rich knowledge about brainstem neuroanatomy, all the pathways that go through the brainstem. Any introduction to neuroscience textbook is going to devote a big proportion of that textbook to brainstem neuroanatomy. And because of this, maybe because of the impression that we know so much about this wonderful structure, I really love to ignore the brainstem, because why not look at the cortex where things are actually fun and interesting? Don't worry guys, I'm looking at the brainstem, so I'm allowed to insult it every now and then. Now the thing is, when you start to think about why is it that we know these things about the brainstem, a lot of our knowledge is coming from animal studies. It's coming from ex vivo histological cell staining studies. And so what this means is that there's a general bias of our understanding of brainstem functional neuroanatomy towards functions that are conserved across species, right? So when we look at the anatomy, we tend to see things in other species. When we look at the function, we're looking at other species. And so as a result, we don't actually know very much about what is going on up in the brainstem in humans that are both alive and awake. Now, because I am guilty of being a little bit corticocentric, I am going to kind of, the main question at any rate in this project is really, what does in vivo brainstem function tell me about cortical function? So when I extend my perspective of cortical function to include the brainstem, how is that going to change the way that I understand all these cortical organizational patterns that we observe? So of course, it's difficult to measure resting state dynamics in the brainstem. It has lagged behind the cortex for many reasons. One of those is that we need really high spatial resolution and high sensitivity. This is a very small structure. The nuclei are extremely small. And we're also going to need all sorts of physiological noise reduction pipelines because, as you probably know, the brainstem is sitting right next to both CSF and vasculature. Did you know that every time your heart beats, your brainstem wiggles? So just imagine what a pain it is to try to extract any nice signal from this structure. And maybe a smaller point, but still really important, I think some of you will appreciate this, we need some sort of atlas or template, because if you want to study all these different nuclei in the brainstem, it is a huge amount of work to try to delineate all of these different nuclei by yourself for every single subject. Really what you're going to do is you're going to say, well, I'm interested in one nucleus, I'm going to delineate that one and leave it at that. But what's really nice is that there are people out there like Marta Bianchardi, who is a professor at Harvard, and she has spent more than 10 years really delineating these different brainstem nuclei in template space. And these are available for anybody, so you can take your brainstem and then parcellate it according to these different nuclei. And not only that, she's really a brainstem imager expert. She then, in 2022, put out these resting state functional connectivity data between brainstem nuclei and cortical regions. This is brainstem specific or brainstem optimized acquired data. So if you were just to take, let's say, HCP data and try to go into the brainstem, you'll probably notice that the signal is not very good. It's just quite noisy. But depending on your acquisition, so in their case, what they're doing, if you want to know the details, acquisition of the head coil has been tilted back a little bit to improve the signal in the back of the brain. And then, of course, there's all these sort of extra noise reduction steps that they've implemented into their preprocessing pipeline to try to optimize the signal in this structure. So here's the brainstem. This is what I'm working with. This is 58 nuclei. I'm going to be plotting it for the most part as node centroids. It's just a little bit easier to see. So this is a coronal section, a sagittal section, and an axial section. It's, again, 58 nuclei. I'm looking at 400 cortical regions, but I will be replicating all of this in, I think it's a sample of 200 or maybe 100 cortical regions. And it's a pretty small subset of people. It's only 20 people, because, of course, this is a really detailed kind of specific data set for the brainstem. But a lot of this also replicates to other data sets. So one of the first things I do with the brainstem is I just want to validate that those nuclei are where they should be. And so what I did is I took these different PET-derived neurotransmitter receptor density distributions across the brain. Now, PET is nice because it's not supposed to show too much of an SNR drop-off in lower regions the way MRI does. And so what we did is we just parcellated these receptor maps to these different brainstem nuclei, and we just double-checked that it looked right. So for example, the serotonergic receptors are more expressed in the RAFA nuclei. We see norepinephrine in the locus coeruleus, these kind of things. So I'm really going to be focusing on this functional connectivity matrix. So for those of you who don't know, this is looking at the synchrony of the bold time series between brainstem nuclei and cortical regions. And I'm going to be focusing on brainstem to cortex connectivity, which you can see already is quite low compared to cortex functional connectivity, probably because the SNR is lower here. But if we zoom into cortex brainstem functional connectivity, we do actually see these stripes in the data, both horizontal and vertical. And so what this means is that there are these dominant or recurrent patterns of connectivity between the brainstem and the cortex that are showing up over and over again. So what are these patterns? Here I'm taking a look at, for every brainstem nucleus, I am scaling the size of that nucleus and the color of that nucleus according to how functionally connected this nucleus is to the whole cortex. So it's essentially a weighted degree map, and you can see, you can look for your kind of favorite brainstem nucleus, but what I like to see when I look at this is take a step back and sort of blur, squint a little bit, and I try to imagine if I wanted to make a network that was very efficient to travel along, I would be putting the hubs at the top, at the middle, and at the bottom, and that's what we see here. We see that the nuclei in the brainstem that are most connected with the cortex, they're occurring at the top, at the middle, and at the bottom. Now on the flip side, you can kind of look at the other perspective, which is how functionally connected are different cortical regions with the entire brainstem, and here we get this really nice anterior to posterior gradient, showing me that these anterior cortical regions are much more functionally connected to the brainstem than the posterior regions. This aligns quite well with the cytoarchitecture of the brain, so depending on the cell type distributions of a cortical region, you're going to show a different functional connectivity profile down to the brainstem. Now, my actually favorite finding when it comes to this anterior to posterior gradient of cortex to brainstem functional connectivity is that when I saw this, it immediately reminded me of these distributions of power of oscillatory rhythms in the brain. So as you can imagine, the brain oscillates, these neuronal populations are oscillating at different frequencies, and depending on where you are in the brain, some frequencies are going to show really high amplitude, so high power, and in different regions you'll see different profiles of oscillations. And it turns out that all these oscillatory patterns in the brain are actually quite correlated with how much these cortical regions are connected to the brainstem, suggesting possibly a role of brainstem input modulating these oscillatory patterns. This is coming from MEG data. Okay, so the main question that I was interested in asking when I got this data was I wanted to know if it's the case that I can cluster brainstem nuclei in terms of the way that they are functionally connected to the cortex. So in other words, can I put together all these different brainstem nuclei and find that there are some sets of brainstem nuclei that are connected to some specific networks, let's say in the cortex. So here we have a brainstem nucleus by brainstem nucleus similarity matrix. So this isn't functional connectivity. Functional connectivity would be the correlation of the time series. This is a bit different. This is telling me for every 2 brainstem nuclei, how similarly are they functionally connected to the whole cortex. And I've clustered it, and you can already see from the clusters that you get these nice groups of brainstem nuclei that are all similarly functionally connected to the cortex. Here I've colored the brainstem nuclei in terms of their clusters. This isn't the only clustering solution that looks nice. There are a couple others. That's what that little dendrogram kind of looking thing on the right is trying to show, how these clusters get combined depending on the scale of this analysis. But the point is that we can put together these brainstem nuclei, we can kind of cluster them in terms of how similarly they are functionally connected to the cortex, and we get these groups of nuclei, which for me, before doing this project, I was not very much of an expert in brainstem neuroanatomy, and so when I look at this, I was sort of like, okay, well, I get some, colors in some brainstem nuclei, but I don't really know what this means, and so my instinct was, instead of focusing very much on the brainstem, to focus on the cortex. And so here what I'm doing is for every single one of these clusters, let's say the yellow cluster, so that's all the yellow colored brainstem nuclei, I'm going to sum up how all those nuclei are functionally connected to the cortex. That would be this pattern right here. So all these yellow nuclei, they're functionally connected very similarly, all very similar to each other. That's how they got clustered together. They're all functionally connected to this yellow pattern in the cortex, which, by the way, if you look at it, is actually very recognizable. This looks quite a lot like the sensory motor, right? Like sensory motor functional activation patterns. Now we do this for all the different brainstem nuclei. We get all these different functional activation patterns in the cortex. And this was really exciting when I first saw this because it's really not noisy. We get these really nice functional activation patterns that are quite recognizable. We get these patterns that look like a sensory motor activation. We get the pink pattern that's very limbic, showing up in singular regions. And we get a secondary motor activation pattern that's the gray one. And then, kind of fun, the green and the blue networks are quite transmodal. And you don't have to just take my word for it. I did a neurosynth decoding where I'm looking at the different terms, different brain regions that light up when people are in the scanner doing these different kind of task paradigms coming from neurosynth. So this is a probabilistic method for associating brain regions to the functions that they might be involved in. And indeed, we get that these different cortical networks, which again, these are connected to very specific modules or groups of brainstem nuclei, they're all related to very specific domains of psychology, including higher order cognitive processes, which is not usually something you would think of when you're thinking of brainstem function, right? Now then, of course, the question becomes, well, why is it that these brainstem nuclei are functionally connected to those cortical networks? This is pretty data-driven, right? So I've kind of shown you some clusters, but what is it about their biology that gives us this relationship? And the intuition or the hypothesis maybe is that surely this has to do with something with the neurotransmitter receptors. Because as we know, lots of these brainstem nuclei, they are synthesizing the neurotransmitters, And then all these receptors are distributed along the cortex, and so you might imagine if you're sending some signal from the brainstem, it's going to affect cortical regions differently depending on the receptors that are expressed in those regions. And so indeed, here what we're doing is I'm mapping 19 different neurotransmitter receptor density distributions in the cortex. I'm using those receptor density distributions to try to predict that cortical functional activation pattern that is associated with the brainstem cluster. And what I'm finding is that there's this three-way relationship between the receptor that is involved in the cortical network, which is related to the brainstem cluster. Now that's a bit confusing, so let me try to unpack that or give you an example. Here, the dopamine transporter. This is the receptor or transporter that is most active or most contributing to predicting the green cortical activation pattern. And the green cortical activation pattern is related to those green brainstem nuclei. That's where the substantia nigra is synthesizing dopamine. Same thing for norepinephrine. We have the norepinephrine transporter being the most dominant towards the blue network. That's where the locus coeruleus is. This is what's synthesizing norepinephrine. So it seems like there's this kind of nice three-way relationship between the neurotransmitter that's synthesized in these brainstem nuclei and then the receptors that are expressed along the cortex, giving rise to these functional activation patterns. Now, if you remember back then, I was taking a look at how similarly are two brainstem nuclei in terms of the way that they're functionally connected to the cortex. Now I'm going to flip that. I'm going to say how similarly are two cortical regions in terms of the way that they're functionally connected to the brainstem. So here we have, again, this is not functional connectivity. This is just a similarity matrix, region by region similarity matrix, telling me how similarly are two cortical regions functionally connected to the brainstem. And this I did not cluster. I know it looks like it's been clustered, but I didn't do anything to it. I was using the Schaefer parcellation, which is, as you might know, already ordered according to the Yolk-Greening resting state networks. So as soon as I made this, I looked at this and I thought, well, okay, this is definitely the Yolk-Greening resting state networks. I know what's coming out here. If I take the first gradient of this matrix, I get this very kind of commonly found or putative unimodal transmodal axis. Now what's kind of fun about this axis is if I correlate this with the version that I get from just cortical-cortical connectivity, this one is actually quite a bit more bimodal. So that makes me think that maybe there has something to do with brainstem input kind of seeding the two extremes of this gradient and cortical-cortical connectivity kind of blurring the picture in between. Usually we think of this gradient as representing graded changes in cortical-cortical relationships, not so much cortical-brainstem relationships, but I wonder a little bit if the brainstem is involved in kind of setting the stage for the two poles of this axis. So then where are these cortical regions functionally connected? I find that these unimodal cortical regions are more functionally connected to caudal brainstem plus the inferior colliculus, and that the transmodal cortical regions are more functionally connected to the rostral brainstem minus the inferior colliculus. So altogether, this is kind of a whirlwind summary showing you that we can take the 7T fMRI brainstem data set and the brainstem atlas to extend our perspective of the cortical connectome or the cortical functional connectome into the brainstem. And we find these hubs that are representing strong connectivity between brainstem and cortex. We find clusters of brainstem nuclei that subserve very familiar cortical networks that are involved in very specific domains of psychology and cognition. This relationship between the cortex and the brainstem is likely mediated by these neuromodulatory systems, the neurotransmitters that are synthesized in the brainstem, and then the receptors that are expressed in the cortex. And lastly, this unimodal transmodal cortical hierarchy, which we typically think of as representing cortical-cortical connectivity. It's actually also found when you look at cortico-brainstem connectivity, and it reflects patterns of functional connectivity with rostral and caudal brainstem. So in other words, kind of one sentence summary is that a lot of these cortical organizational patterns that we see in the brain, cortical function, things like neural oscillations, the cellular types, the different cognitive specialization, a lot of these can be traced back to the brainstem. So thank you for listening. This work is published. Code and Data is on GitHub. I do have a poster tomorrow, but it's not about the brainstem, but it's still cool, so you should still come. And yes, thank you.
Amazing, Justine. We have time for questions, so please come up to the mic if you have questions. Okay, I have a quick one about brainstem to cerebellar connectivity, so that might be a logical next step, I'm assuming.
Yes, brainstem, yes, it is a very logical next step that I have not looked at. But yes, it would be really cool to look at brainstem cerebellum connectivity. I think the data is out there, and one of the biggest challenges has to do with trying to know how to analyze data that is coming from very different parcel sizes. Some of these brainstem nuclei are really tiny, and then some of the cerebellar regions are like really massive, depending on how you apart. Like cortex, we can sort of divide into equally sized regions, but in the cerebellum is typically more functionally or like anatomically defined. I think that's the biggest challenge. But yes, absolutely. Thank you.
Thank you. So let's thank the speaker one more time.